Hi, my name is Dave Champlin. I'm a biology professor at USM, and this is a short video about micropipetting skills in cell and molecular biology. And the first thing is that we almost always wear gloves when we're micropipetting, and most of the time it's to protect our samples from us. And most of the time when we're talking about samples in this setting, it will be DNA, RNA, like that, and in the environment from formerly living cells is dust that includes, the carcasses of old cells from our skin and every other kind of organism. And there are some enzymes in those, and some of the enzymes that degrade DNA and especially RNA, ribonucleases, are really stable. And so when we're working with DNA and RNA, we're concerned about the dust in the air and just the materials on our fingers. So we wear gloves to protect our samples. And then the next rule is that we would just keep things covered. So we'd open this up to get a tip, but then we're just going to cover it up. And that's going to minimize that dust, that contamination that could destroy our samples, like that. And so that's the first habit of cell and molecular biology micropipetting, is that everything stays covered, like that. We want to keep it clean. Then here's some micropipettors, and there are a variety of different brands, and they all have the same features. And the first feature to mention is that the gears inside of these are always made of plastic. They could be a super premium micropipetter, and the gears are plastic. There is a way near the handle to adjust the volume and turn those gears. And in turning the gears, you want to just keep in mind they're plastic. And if they ever jam, which they sometimes do, you want to be gentle and sort of toggle things back and forth and try to release them, and not just crank it. Because if you crank it either in a jammed micropipette or at the end of its limits, you'll just destroy the plastic gears, like that. So, like I said, there's a way of adjusting the volume. So every micropipetter has got a volume control. Well, some are fixed volume micropipettes. Those are pretty unusual. Most of them are variable volume micropipettors, and there's a volume control on the side. And then there'll be a numerical indicator. And there are ranges for micropipettes, and so here's some examples. Typically, ranges are written on the side of the micropipette. And this is a pretty classic range of 3 micropipettors, and one that is great at measuring volumes up to 20 microliters, and then one from 20 microliters up to 200, and then one from 200 up to 1,000. And the idea is that this one is not as accurate under 200 as this one, and so forth. So you always want to try to pick a micropipetter that's appropriate for the size. There are other sizes. Here's one for really small volumes, and so forth. But those are the classic 3. And the next thing is that the numerical indicator is relative to the volume. So for example, this one has got a little yellow line, and that marks the decimal point. And we know that's the decimal point because this is going to measure something around 100 microliters, and there it is set for 100.8 microliters. Here's one. The decimal point is here, yellow line marking this. This one measures between 1 and 20 microliters. It's set for 4.36 microliters. This one measures up to 1,000. There's actually no decimal point here. This would be 935 microliters. So that volume is always based on the size range of the micropipetter. Okay, next thing is that there are different tips. We always use a disposable tip, and there are 2 classic tips for micropipetting. These tips are often called yellow tips because historically they were yellow, and these tips are blue. And the blue tips are used for 1 milliliter or 1,000 microliters. Those are equal volumes. And so we got blue tips for the larger volumes. Boom, like that. You just tap them on, and they're disposable. So I tapped it on, I took the lid off, I put the lid right back on. And then we have yellow tips for these smaller volumes, like that. And then there are a variety of specialized tips, sometimes for a smaller volume micropipetter like this one, boom, little tiny tip. Sometimes the tips will have a little cotton in them. They're called aerosol resistant tips, and this one doesn't have it, but they have a little cotton tip in there to protect the liquid from contamination. That's the next thing to mention, and the tips are clean and disposable, it's understood that the micropipetter itself is dirty. So the tip is dirty and the shaft of the micropipetter is dirty. And that brings up a couple of points, and one is that when you're micropipetting, you want to very slowly draw the liquid up. First, so it measures accurately. Second, so it doesn't splash. You don't want liquid to splash and touch this shaft of the micropipetter inside of the tip. And you want to be really careful with that. And we'll talk some about that. If it happens, it's considered contaminated and bad. And that can happen either by splashing things up, by lifting stuff up too quickly, or by tipping it. So when you have a volume in a micropipetter, you don't want to tip it like that. Sometimes you think, oh, I got to get the cap back on this tube, but you don't want to tip a micropipetter up like that when it has liquid in it. Okay, then the next thing is, if the shaft is dirty, you also don't want to do this when you're micropipetting. You can put the tip, clean tip, into a bottle or test tube like that, but you don't want to put the shaft in to the tube. How can I pipette out of here? And the answer is that scientists, if they had a large volume with a small volume, is they would get a smaller tube and they'd pour some out into that smaller tube so they could safely micropipette out of this. Then another thing they'll do is they will often tip the container like this and tip it so the liquid is up near the top, so they don't have to push the shaft into the vial, tube, or bottle down to the point where this is in and could contaminate it. Again, you know, it's clean. We could try to clean it up, but nevertheless, it'd be considered dirty and the disposable tip clean. So scientists will often angle their pipetting like that, so that only the tip goes into the vial. Next thing is that, for pipetting, is that we are first going to set the volume. So we set the volume, then put a tip on, and the tip is sort of the last thing we'd put on. We want to keep this clean, so we'd put it on last when we're ready to pipette. And then we'd remove the cap of our tube. Again, keep these covered like that. And then when you push down on the button, you can push really hard, and it comes to a point where it won't move in any further. That's not how far you want to push when you're measuring the volume. There's a first stop, a gentle stop, and that's where you stop when you're going to measure. So the displacement between the rest and the first stop is the measured displacement that you're going to use. So the way to pipette is to get your bottle ready, and then before you put the tip in the liquid, you gently depress to the first stop, no further, hold it, and then you put just the tip in. You don't want to plunge the pipette deep into the liquid, because the pipette has some gaskets that can wear, and the pressure, kind of the pressure of the liquid, could cause the tip to fill. I don't know. Let's see if I can do a little bit of that right now. So if I push the pipette in far, if it's an older pipetter, some liquid would start to fill into the tube, and we don't let that happen. So it's appropriate to just put the tip in and then withdraw liquid, lifting up slowly until your hand is at rest. You don't want splashes to happen like that. Then you want to have the tube that you're going to dispense it into nearby, because as you move things around like this, you're accelerating the tip and the liquid could kind of pull out. Or some liquids don't have just water in them, and they have less surface tension. So some liquids are more sort of drippy. And so it's really good to have the tube that you're going to dispense into, and I'm just going to dispense back into here. And when you're dispensing, you just want to release the tip, release the volume smoothly like that. And now is when you can push the plunger down further past that first stop to make sure that you get everything expelled like that. The next rule for good pipetting is that when we're micropipetting, we often are micropipetting multiple volumes into a target tube. So a little of this, a little of that. And what we want to do is we'd like to pipette in a first volume that's a big volume, and then pipette smaller volumes into the big volume. So for example, let's say we have a tube, we're going to put 100 microliters of water in it. Let's put the 100 microliters of water in it, and then we'll put in the smaller volumes, maybe 10 microliters of a buffer, maybe 1 microliter of an enzyme, like that. Because the small volumes, we'd like to, instead of just squirting them into the tube, we'd like to eject them directly into the target volume. So we'd like to add 100 microliters of water, and we can do a little of that right now. So let's just to show you, pipette the first volume, the big volume first, and here's another thing. I shouldn't have gotten that tip ready because I'm not ready. And here's another thing. So we're going to open this up. We kept it covered. These are tubes, and rather than putting our hand in there, even though our gloved hand is relatively clean, we also now, because we've been wearing gloves for a while, we consider our gloves to be kind of dirty. So we wouldn't just put our grubby gloved hand in there. And instead, it's good to spill a couple of these out, and then pick one up without touching the other ones. You can pour the other ones back in, like that, and then it's good, if you're not going to use it right away, to cap it. Maybe you want to label it. It's always good to cap it and label it. And then when you uncap it, these have a little thumb flange, and you want to try to just use the edge of your thumb. Lots of times the gloves will be sort of loose on your fingers, and it's good to pull them back. It's good to wear gloves that are the appropriate size, so they're kind of tight on the tips of your finger, so that you can open this up by opening it like this and bending it back. This hinge is plastic, so you actually have to actively bend it back to get it all the way, and you try hard not to touch that inner part of the cap or the inner part of the tube. And so, boom, like that. And these are disposable tips, and so it's pretty common to just get rid of them, right? You weren't ready to use it, so just throw it away. And we use quite a few disposable tips. So here's some liquid. We'll pipette this first. Take the cap off. Set the cap down like in that position, because we're going to keep the inside clean and the cap part dirty. Tip this at an angle. Push this down to the first stop, and try not to put the pipette shaft in. And then, next thing I haven't mentioned is you want to watch yourself pipetting. So I kept this up close to my face so I could watch the pipetting to make sure that I didn't get any air bubbles, and that I didn't splash, and that I got the approximate volume I need. So over time you get used to seeing this and saying, yeah, it looks like about 100 microliters. I'll put the cap back on, and it's very nice to have that. So you always want to, if possible, to lift tubes up and watch yourself pipetting in, and then watch yourself pipetting out. You squirt it out, then you push down, you get rid of all of it, get rid of that tip, and we could cap it like that temporarily, because now we're going to put a smaller volume in, like this. So let's say we're going to put 4 microliters in, and it's good to eject the 4 microliters into the bigger volume. So let's say we're going to do this, and uncap this, and push this down to the first stop, and then just gently lift up. Again, I'm watching myself doing that. As I pulled out, I kind of wiped it on here to wipe off any liquid that's on the outside of the tip. See how I'm turning this really gently, because I do want to put the cap on, but I also want to pay attention to the liquid that's in there. It's a really small liquid volume, so it's not likely to fly out. A larger volume, I wouldn't do that with. So we got this small in here. I'm going to uncap this, bend the cap out of the way, and then I'm going to look at this and watch as I put the tip into the liquid and eject the liquid, and then I'll plunge it up and down repeatedly and rinse it out, and then I'll use the final push to get everything out of there, and I've mixed my small volume in with my big volume. That's really good. And then the final thing I just want to point out on here is that often with these little tubes, we'll give them a spin in here, and the convention is that we'll put the hinges out, pointing out. And so we'll spin in a centrifuge. We need to have the tubes balanced across from each other, and we'll put the hinges out, and then this little cap pops on, snaps on like that, and snaps on for this centrifuge. And the reason we put the hinge out is because this is called a fixed angle rotor. So tubes like spin like this, And instead of spinning stuff down to the bottom of the tube, if there's material that's going to spin down, it will spin down and make sort of a comet tail shaped mark along the outer edge and bottom of the tube. Lots of times that material is stuff that we don't want to touch. Sometimes it's debris that we don't want to touch. Sometimes it's our product that we don't want to touch. So sometimes it's cellular debris, and we're going to pull the liquid off and save the liquid off, clean liquid away from the debris. Other times it's a DNA that we precipitated, and it's in a pellet. We're going to get the liquid off and discard the liquid. After centrifuging, we talk about the pellet, and then the liquid is referred to as a supernatant. And the reason we put the hinges out is because sometimes the pellets are really small, and we want to avoid them when we're taking off the liquid. And the way that we do that is we will hold the tube, we'll open the tube up, and we'll hold it with the hinge up, and we'll use the other wall, inner wall of the test tube to steady our hand and slide down the other wall. We know we can do that safely because any pellet is on the hinge side. So in other words, it's in the upper side. So we're going to slide down the inner wall of the lower side of the tube, and we can start withdrawing the liquid as far away from the pellet as possible. And because the pellet is not at the bottom, it's really more on the side wall and down near the bottom, we can actually withdraw all the liquid by going just to the bottom of the tube, and we won't bump into either debris or product, and we can get rid of we can take out all the liquid and either save it away from the debris or discard it like that. And so that's the last thing I want to mention in this video about centrifuging test tubes of this type. Okay, that's it for now.